What we're going to do this morning is point to the future. And I'm going to introduce John Epstein, who's been on the faculty for a number of years. He's trained as a cardiologist. He's the former chair of our Cell and Developmental Biology Basic Science Department. He's launched a number of our centers and institutes and really helped to innovate a number of programs here at Penn Medicine. He's currently serving as the Executive Vice Dean and Chief Scientific Officer, and will introduce you to some of our star faculty who will point the way to the future. John, the podium is yours.
Thank you very much, Larry, and it's my real honor and pleasure to help to welcome everybody here for the Alumni Weekend. It's a pretty special time to be a faculty member or a student or a staff member here at Penn Medicine, and I hope that while you're here, you can get a sense of the palpable enthusiasm and excitement that really pervades the place these days. Larry mentioned the number of FDA approvals. The number of new basic science discoveries that are being published in high-impact journals has been skyrocketing over the last couple of years. And the coordination and collaboration between the very basic scientists, the students in those laboratories, and the clinicians who are, delivering just outstanding care in this region is a special and unique thing about Penn Medicine. And we're all very proud to be working here these days. I'll just convey one story that I've told before that I think conveys that as well as anything. Last summer, when we got FDA approval for CAR T therapy for cancer, Bob Vonderheide, one of our panelists, organized a flash mob in the lobby of the Perelman Center for Advanced Care that was absolutely packed. Everybody showed up, from the Abramsons, the president of the university, the physicians who were doing the work, the scientists, but also all the staff members. And it was a real moment of excitement, of shared enthusiasm. And as I walked out, I went past the fellows parking the cars outside and said to one of them, pretty exciting day. And they said, "Yeah, Doc, I love coming to work every day because I'm part of curing cancer." But, I thought if that's what the folks parking the cars are thinking, we're doing something right. And I think that is the sense of shared experience that we're having here right now. So we have three just terrific panelists, three institutional leaders who I think exemplify a lot of what's great about Penn here today. They're each going to give a very brief introduction with a couple of slides to tee up some questions. I'll ask a few questions myself and open it up to the audience so you can ask some questions as well. And I think what you'll see is that their presentations and their backgrounds and interests epitomize what I just said about the balanced portfolio at Penn and how we're making really great advances in all aspects of that portfolio, from very basic mechanistic science at the molecular and nanoscale to the types of new therapeutic opportunities that are going to be taking place in this beautiful new hospital that's going up next door here. So our first speaker is Kristen Lynch. Kristen is a professor and chair of the Department of Biochemistry and Biophysics, and she's an expert in RNA biology herself. She's a role model and a terrific institutional player, and I was pleased that I could talk her into taking the job as chair just a couple of years ago. She did all her training at Harvard and at UCSF. She went to UT Southwestern to begin her faculty career, rose to the level of associate professor there before we were lucky enough to recruit her here to Penn in 2009. And she's been an energetic and forward-thinking leader of one of our most basic fundamental science departments. And I'll invite Kristen to make a few opening comments.
Great. Well, thank you, John. And it is indeed a very exciting time here at Penn Medicine. And I feel very privileged today to be able to, on behalf of my Department of Biochemistry and Biophysics, to tell you a little bit about how the basic scientists in my department have been able to contribute to, as well as leverage and benefit from, the revolution in medical care that is going on here at Penn. So one question one might ask is, why at a medical school should you have a department of biochemistry and biophysics? And for many of you who were here as medical students, the word biochemistry may conjure up fears of that first-year class that is sort of the bane of the existence of many a starting medical student. And other people might reasonably ask whether the fields of biochemistry and biophysics are perhaps too theoretic or too distant from medicine to really have impact on patient care. And what I hope to convince you of in the next few slides is how leveraging fundamental biophysical technologies and insights and combining that with fundamental questions in human biology really can have profound impact on human health. And that's the vision that drives our department and the mission, the stated mission of the department. And we feel very fortunate to be here at Penn, where the administrators and the leadership share that sense of vision. And what that means for us is that here at Penn, we find both the support for the infrastructure, indicated here by many of the toys and facilities we like to play with as biochemists, but also supporting the intellectual infrastructure, where we're challenged and encouraged to ask fundamental questions and then to partner those with the clinicians to really drive forward medical advances. So today I want to talk about two specific technologies that has really captured the interest of our department. The first is electron microscopy, and the second is protein studies, or studies of protein chemistry. And then I'll end with just some very short vignettes on how we're using technology really directly in terms of benefiting or impacting patient care. So let's start with electron microscopy. This is a tremendous revolution that's going on across the biomedical field across the world. And indeed, the technology of electron microscopy was awarded the Nobel Prize just this past October in recognition of how transformative this is. In essence, what electron microscopy allows us to do is to freeze samples, so capture biologic samples, and then look at them with electrons to see at a level that even the highest light microscopy can't get at, so that we can really get pictures, get snapshots of biologic molecules in action, so as they're carrying out their activity. So what does that mean here at Penn Medicine? So one example in which we've used electron microscopy is in the work of a very up-and-coming young professor in the department, Ben Black, who studies this question of chromosome segregation. So as most of you are probably aware, all of our cells have the same genetic information, and when cells divide, either during embryo development or when your blood cells, you need to make new blood cells, the genetic information has to split equally. And the way that's accomplished is through these anchoring points called centromeres that function essentially as tether points or anchor points for these, the ropes, if you will, that pull apart the chromosomes. And when these anchor points are not accurately formed, we get breaks in chromosomes, and genetic information does not segregate appropriately. And this has direct implications both for infertility as well as cancer. So Ben Black is really interested in understanding at a very mechanistic, basic level, what do these anchor points look like? And if we understand how they look, maybe we can understand how they go wrong in disease and how to improve that. But these are complicated little features to study. They're not a single entity. They're a complex assembly of proteins and DNA all wrapped up together. And through modeling and sort of piecing parts, we've been able to come up with some general ideas of how this looks. But really drilling down and understanding at a deep level has been hard. Another example of the use of electron microscopy is its ability to capture multiple different states. So many proteins that drive all of our biologic processes are not static. but they need to move and they need to carry out some activity. And through electron microscopy, we're able to capture many intermediate states and then put together a movie very much alike, similar to the old-fashioned movies where you flip through the many pictures. So Jim Shorter has been using this to understand the process of protein deaggregation. Most neurologic diseases, Parkinson's, Alzheimer's, ALS, have been recently recognized to be due to the fact that proteins, these machines in your cells, get balled up into a tangled mess, very similar to sort of a junk heap where you can't separate out the individual machines from that pile. So how can we accomplish, or how can we prevent the sort of aggregation that causes neurodegeneration? Jim has identified a protein, HSP104, that acts to tug on the ends of these tangles and unwrap them and allow for cell viability. And indeed, in animal models, when he expresses this, he can reverse neurotoxicity and promote neurologic function. So using EM, there we go, he's been able to capture not just a static picture of this machine, but actually look at it pulling on the proteins, tugging on these tangles that are causing Alzheimer's, that are causing Parkinson's, and releasing the protein to be no longer toxic. And you can, this is just an amazing movie, watching it walk up the protein, and you can see very directly how this has to happen in the cell and how we could leverage this to promote human health. So, but looking at a protein is not the only thing that's important. We also need to understand its chemistry. And when most of us think about a protein interacting with, say, a drug that you may use to treat one of your patients, we think about how the drug interacts with the protein. What are the bonds that are made? What are those interactions? But it turns out that's not the only thing that drives whether or not a protein and a drug interact. It also is important to understand how the protein wiggles or how the drug wiggles. Because if you think back to thermodynamics, binding is a sum of both the interactions and the change in motion. So Josh Wand, another faculty in the department, has developed a method to study how proteins wiggle. This is something that people really haven't been able to look at before. But using NMR, nuclear magnetic resonance, he can see proteins wiggling and see whether they change more or less, whether they wiggle more or less when bound by a drug. And he's now using this to ask whether you can use this to improve drug development. And indeed, if he staples a portion of the protein together so that it no longer wiggles, he can now increase the ability of a drug to bind to the protein, to inhibit the protein function by over 500-fold. And very excitingly, we've just partnered with a local pharmaceutical company to get a higher-end NMR machine. And on Monday, I've been told that Josh and this company are going to start experiments in leveraging this technology and this breakthrough to see if we can improve drug development. Another form of protein chemistry is modifications that can happen on proteins. And recent advances in technologies such as mass spectrometry allow us to really look at the chemistry of the protein and understand what that protein is and how it's modified. And we're tremendously fortunate here at Penn to have really one of the rising stars in this field of looking at protein modifications through mass spectrometry in Ben Garcia. And what I'm showing here is just some of Ben's many impactful studies where he's taken tumors, and this is from mouse, but it's a good model for a human tumor, looked using mass spectrometry at how those proteins are modified in the diseased muscle versus the healthy one, and has identified modifications, chemical modifications that are attached to the protein that alter their protein function. And in understanding how the disease tissue has different modifications than the normal tissue, he could make the very immediate prediction that if you control the level of this modification called acetylation, that might return the tumor back to something that looks normal. And indeed, there are inhibitors that can control this acetylation process. And in mouse models, they can show that if they use inhibitors, two enzymes that control acetylation, that you remove tumor growth in this mouse model. So again, something where you could really directly see how understanding the details of the protein chemistry can impact patient care. So what about more direct impact where we're currently using technology to think about patient care? One example of that is work from Sergey Vinogradov, who's a synthetic chemist in the department. The department really ranges from biochemistry all the way to chemistry. And Sergey has used his knowledge of chemistry to design these complex molecules that are able to fluoresce in the presence of oxygen and are also bioavailable. So these are now being used worldwide. They can be injected into synovial fluid, into blood, and are being used, as shown here, to image blood vessels. They can also be used even in live animals to look at changes in oxygenation in response to stress or in response to various chemical drugs or other compounds. And that's shown here, looking at electrodes coming off of a mouse showing hot spots of oxygenation and low oxygen. And this is being used, as I said, even already in patient care to look at changes in oxygenation in response to stroke, in diabetes, in cancer, et cetera. Going back to Ben Black's work, as I said, he was interested in chromosome segregation. When chromosomes make breaks inappropriately, they need to be repaired. And one of the enzymes that does that is this enzyme PARP1 that basically forms a gel along the DNA that serves as a Band-Aid and promotes reassociation. PARP1 has been used, as I think John or Larry pointed out, as a target of anti-cancer therapy, including the new approval here from the Vassar Center. But not all PARP inhibitors seem to work. And Ben was able to use the same sort of methodology I just described, looking at mass spectrometry, looking at how the protein wiggles, to understand actually and be able to predict which PARP inhibitors are going to function and block the activity and cure cancer and which won't. And so he's very excited to continue to work with the clinicians here to push that forward. And then finally, work out of my lab, we study another way that genes can be put together and taken apart, and that's called alternative splicing. And in essence, all of our genes are made of multiple building blocks. And the cell takes advantage of this by including or excluding certain blocks depending on cellular need. And normally in our cells, that's leveraged by our body to encode different proteins, different machines, according to our need. However, sometimes this can be problematic. And as you'll hear, I'm sure, in the next talk, we've made fundamental successes here in CART19 therapy, where we engineer T cells that are targeted against epitopes on leukemia and help to cure cancer. But it turns out that as much as this is a wonderful transformative technology, there have been a few patients that have proven resistant and have relapsed with tumors regrowing. And was discovered here is that in these regrowth or outgrowth tumors, the cell had chosen to skip the building block that encoded the part of the protein that the CAR T cell was directed against. So in essence, the cancer goes stealth and is no longer visible to the therapy. And the work that my lab does in terms of understanding the mechanisms of this has allowed us to now be part of a multi-team group that are working together to understand how we can alter this to allow these relapse cancers to now be sensitive again to the CAR therapy. So in sum, why I am so excited about being here at Penn is the fact that really fundamental basic technology is supported and really cutting-edge clinical care is supported. And most importantly, what's supported is us talking together. And it's really through these interactions that happen in the hallway, that happen at meetings, that happen through various mechanisms, formal and informal, that we really have unlimited promise for the future. So welcome back, and I hope we continue to make you proud.
Thanks, Kristen. It's a terrific way to kick us off. I want to just emphasize that I've grown to believe that the space in which we interact makes a big difference. We've got beautiful spaces here, as you know, and I want to thank those of you who've supported us in building those gorgeous spaces. It helps people to interact. It helps our students to interact. And many of our best collaborations happen around those interactions led by our students. Our next speaker is Mitch Schnall. Mitch is the chair of our Department of Radiology. He's an international leader in translational imaging, and he'll take off on this concept of imaging. Mitch is sort of a wunderkind. He's been at Penn quite a while, got all his degrees here. He's brought in, I think we counted up well over $120 million of research support for his own laboratory over the years that he's been here. He builds the infrastructure of the entire school, even beyond the department. He runs an outstanding clinical department. And on the national scale, he's played enormous roles in the types of screening we do for cancer in patients and the guidelines that direct that process. He's a member of the National Academy of Medicine, elected in 2012, and he was awarded the gold medal from the International Society of Magnetic Resonance Imaging in 2013. Mitch is one of our great institutional leaders. Mitch.
Thanks, John. Next time I get an introduction from you, I got to make sure my mother is present to hear about it. So just to amplify some of the comments that Kristen made of the importance of basic science into driving clinical care, a little secret today. I'm playing hooky for the hour because I'm actually assigned to be reading MRI scans today. And the techniques that we're using today on these patients were techniques, many of which were being worked out when I was a graduate student way back in the 80s in Kristen's department. And so there's really a palpable translation of these basic methodologies into clinical care. So I want to talk a little bit about some of the things that are going on in imaging today that we think are going to be driving some of the future. Oops. Which one is there? Here we go. So I really want to focus on two frontiers in medical imaging that I think are really driving changes that we're going to see in radiology over the next several years, as well as changes in medicine in general. And the first is molecular imaging. In molecular imaging, we're actually creating what we call imaging probes or molecules that we inject into people, typically with radioactivity. And in this case, you can see tagging uniquely the estrogen receptors in breast cancer metastasis, knowing that they're estrogen receptor positive. The other thing I'm going to try to highlight is what I like to call biomedical imaging informatics, really recognizing that images are data. And here we're showing a close-up of the temporal lobe, an MRI of the temporal lobe, and how we're segmenting out hippocampal subfields. And the hippocampus is really important to study in patients with neurodegenerative disease and Alzheimer's disease, and we can make biomarkers to follow these patients. And so let's talk a little bit about molecular imaging for a minute and the methodology that goes into that, which is really creating a window into inpatient biology. It starts off with a cyclotron that we use to make radioactive isotopes. And in molecular imaging, we tend to make isotopes that are positron emitters because positrons are very easy to and very powerful imaging agents. The most common positron emitter we make is F18. And that positron emitter then gets very quickly, because it has a half-life of about 2 hours, so we have to do all of this very quickly, gets very quickly put into a facility that actually synthesizes the drug on campus. And so we have a large GMP facility and a drug manufacturing program where we make these on campus. And in this case, I'm showing F18 fluorobetapir. And we like to talk about our FDA approvals at Penn. And this is one that sort of slid under the radar a few years ago. And fluorobetapir is the agent that's being used to image the beta amyloid plaques of Alzheimer's disease, FDA approved, used throughout the world, developed by Hank Kung's laboratory here at Penn. Those are then injected into the patient. The patient's placed in the PET scanner that collects those positrons as they leave the body. And then we've got images like you can see on the bottom right, where on the right-hand side you can see all of the red around the cerebral cortex, which is the beta amyloid plaques in a patient who had known Alzheimer's disease compared to a normal age-matched control, which doesn't show those beta amyloid plaques. We've got a whole host of pipelines of various agents that we're working on, from targeting different neuroreceptors, metabolism, and I'm going to focus on this one to the top right, PARP, which is involved in, as we've heard, several important cancer types. And so, as we heard, what PARP does is PARP repairs DNA damage. It is upregulated in a number of cancers and infers some resistance to treatment because they're very active in being able to repair DNA damage. And inhibiting PARP is a therapeutic strategy that's being used in a number of cancers, presumably or primarily ovarian and breast cancer, but others as well. And so we've created a tag that tags PARP. And the other nice thing about this tag is it tags PARP while it's actually bound to the DNA undergoing repair. So it doesn't tag free PARP, but the bound active PARP is what it tags. And so how do we think of using this clinically, and how does it help us take care of patients? Well, the first thing is, if we knew we were looking for a tumor that had an abnormality and was overexpressing PARP through a sophisticated molecular blood test or otherwise, we could use this agent to find and locate that tumor. The other thing is that the intensity that we see from that tumor will tell us the amount of PARP and quantify the PARP overexpression, which could be very valuable for us to be able to determine therapeutic strategy. And finally, we can actually look at clinical efficacy of inhibition. These PARP inhibitors are very, very toxic, and we want to make sure we get the dose just perfectly right. And so if we look to the top right, this is a patient with 2 PARP-producing tumors in the left side of their pelvis. And the bottom right is after they're treated with a PARP inhibitor. We image them again, and of course, now there's no PARP binding the DNA, and we don't see the overexpression of PARP. And we can titrate that so we get just enough dose to make the effect that we want to see. And so there's lots of ways that we can use this for precision therapy. Let's talk a little bit about thinking about the images that we use as data. Believe it or not, every scan that we take is really big data on its own. So every brain MR is about 10 million data points. And we can use those data points in a number of ways. One is to create sophisticated approaches to visualizing structures in the brain. And so we can look at surface rendering of the brain so that we can make it very appealing for neurosurgeons that need to go in and operate to see the surface structure. We can also start to look inside at some of the structure, in this case, the fiber tracts that infer a lot of functional information that we can get from the brain. And so this is used very commonly to guide surgical therapy every day now within Penn Medicine. And you can see how we look at the relationship of the tumor to very important functional fiber tracts in terms of how we're going to operate and how we're going to pass instruments into the brain. And so very important to maintain function while doing surgery, and we do this every day. We do a lot of mathematical modeling to model normal human tissue, and we're trying to use that to help us think about prostheses. And here, this is work by Alison Pouch and Paul Yuskevich, collaborating with a cardiac surgery group, the Gorman Lab. And what they're doing is being able to use some artificial intelligence techniques to automatically extract abnormal valve planes out of echo images that you see to the top left, and then take models of normal valves and map them onto these abnormal valves so we can actually design the valve model that should be put in that particular patient. We're now starting to work with 3D printing technology to think about 3D printing this valve so we can make personalized prostheses for patients. So another use of the images as data and leveraging that data. I'm going to talk a little bit about phenotypic characterization. These are 2 MR scans, and the bright that you see is the contrast that's being taken up showing us breast cancers. And you can see the breast cancers on the right and the breast cancer, they look very different. They're both breast cancer, but they have different phenotypic characteristics. I've seen thousands of breast cancers on MRI, and I see a lot that look like the one on the right, and I see a lot that look like the one on the left. Well, how do we start to take this and do a rigorous quantitative description of what these phenotypes are? And so this is Despina Kontos's work, where what she does is, again, uses some artificial intelligence algorithms to actually extract the tumor out of the image. Then she takes that tumor and she describes it mathematically in a number of ways. Some descriptors are the perimeter of the tumor, the roughness, and the shape of the tumor. Some are looking at the texture inside using some pretty complex math, as you can see. And what she creates is a number of what we call features. Each feature has a coefficient with it or an intensity with it. And if you think about it, that's a little bit like when we studied genetic expression in a tumor, right? We have a bunch of genes, and each one has an amount that it's expressed. So these are a bunch of phenotype features, and each one has an amount that it exists in the tumor. So we can use a lot of the same mathematical framework to think about this. So here is an expression array plot where down on the vertical, these are all feature descriptions, right? And the brightness or the color is the intensity of these features. And on the horizontal, we have individual patients. And what you can see is there's three major phenotypic classes. To the right, you can see a cluster. In the center, a smaller cluster. And these are represented on the top bar by the red, green, and maroon bars. And then to the left, you can see, finally a wide phenotypic cluster of similar expression of phenotypes. And it ends up that these patients were scanned over 10 years ago. We can look at the follow-up. And it ends up that all of the recurrences sit in the left phenotype cluster. And we can see HER2/neu expression, estrogen receptor expression, and other things are scattered randomly across these phenotypes. These phenotypes tell us something about the biology of the tumor. One of the things that we're doing now is we're studying the relationship between these phenotypes and the genetic expression in these tumors so we can understand what the drivers of these phenotypes are. And so finally, one of the things that we're doing is we're starting to put work together with pathology. And as Kristen pointed out, one of the unique aspects of Penn Medicine is that we tend to collaborate across fields very effectively. And so I'm working very closely with the chair of pathology, David Roth, and we're starting to think about putting radiology and pathology data together in an integrated diagnostics framework. And so one way to think about this is think about how a pilot flying a plane has many, many data inputs coming to them and have to make decisions in real time that have grave consequences potentially for a lot of people, both on the plane and in the ground. And they need displays of data, they need analytics to support them. Well, we're thinking about the diagnostics problem the same way, where we have molecular imaging data, we have quantitative imaging data, we have laboratory data, pathology data, and we think about creating a cockpit that's going to allow us to visualize this data in a unique way, have analytics on top of that to support our analysis of the data, so we can very accurately and rapidly come to the right diagnosis and follow patients effectively. Thank you.
I love the precision medicine cockpit. I think that's the term I'm going to remember. Our next speaker is Bob Vonderheide. He's the director of the Abramson Cancer Center at Penn and the John Glick professor. Bob is absolutely terrific, Cancer Center director and a member of the leadership team here at Penn. He's a pleasure to work with every day. He was an outstanding scientist on his own and led an enormous translational research program before he was appointed director. He continues to do that and now has just blossomed as a terrific institutional leader. He's a Rhodes Scholar, trained at Harvard, at the Dana-Farber, before coming here to begin his independent career. Bob, I look forward to your comments.
Well, thanks very much, John, and welcome everybody back to Philadelphia. We're really happy you're here and able to share in this conversation. I became Cancer Center Director in July. It's been a terrific year. Many thanks to John Epstein and Larry Jameson for welcoming me into that position. I've been at Penn for 17 years. It's been a phenomenal year, so much so that I thought I would take a gamble this morning and just see. You know, I know our alumni head to Boston. Is this too soon? Is this too soon? I also worried that either Kristen or Mitch would show this since I was the third speaker, so I had a backup. Men's basketball team at Penn. So, you know, this has been an incredible year at Penn Medicine and certainly at the Abramson Cancer Center, and I thought this morning I would talk to you about some of the achievements we have and then also to key in on our theme this morning about looking forward. We have a lot to celebrate, actually, in cancer. We understand this to be a major health problem in the United States. However, cancer deaths in this country continue a steep decline. In the most recent survey by the American Cancer Society, 1% a year decline in deaths for the last 25 years, and that's not by accident. That's because, as a country, we're focused on it, and a key part of that has been the comprehensive cancer centers. 2.4 fewer deaths in this country because we focused on the cancer problem in all its dimensions for why there's cancer and how to detect it early. Here at the Abramson Cancer Center, we're really privileged to play a central role in driving these new discoveries and, importantly, taking them to patients. We are a NCI, National Cancer Institute, designated comprehensive cancer center. There's about 45 in the country. We have been most recently rated in our core grant renewal as exceptional. That's the highest score. Larry, where's Larry? It's the highest score you can get. And we are, I think our cardinal feature is that we are science-driven, patient-focused. So we combine a massive research organization with clinical care. We have 6 adult hospitals, 1 children's hospital, 290 faculty members doing research. All the metrics for the Cancer Center are going up. U.S. News and World Report most recently ranked as 10. That's the highest we've ever been in. We feel we're proud of that, of course, and Penn proud, as you are, but also the responsibility with this incredible momentum to keep it going. These are our priorities and our values. I'm sure you would agree with them because these are not new values to the University of Pennsylvania. You were familiar with them when you were students here. But to capitalize on discovery, as we've been talking about today, harness our collaborative culture, can't underscore that enough, drive leading-edge new therapies and prevention strategies, I'll get into that in a minute, and lead the new age of precision oncology. You've already heard of many collaborative examples today. The first Cancer Center director was Peter Noll. How humbling for me to think that I might follow in his footsteps. Peter, many of you knew him, won the Lasker Prize, and discovered the Philadelphia chromosome. So when we talk about a revolution in cancer, you don't have to look much further than this city for any types of revolution. Peter changed how we understand cancer and showed that it's a genetic disease. And this started the era of cancer genetics. Now, this goes back 40 years. And in many ways, many of the activities we've been doing around understanding the molecular biology of the tumor cell and finding genetic abnormalities and drugging them all go back to Peter's seminal observation that he laid out here just a few feet away. Well, I would say, and I'm sure Peter would agree, that over the last 5 years, what's happening is we've moved from the view of cancer as just the cancer cell to a new era, and I'll call it the era of the cancer ecosystem. So this is an H&E of pancreatic ductal adenocarcinoma, highly lethal. We still have not accomplished anywhere near what needs to be accomplished in taking care of our patients. And what you can see in the middle, let's see, here's the laser pointer. In the middle, this is the tumor cells, these dark, deep purple cells. But the rest of this stroma is actually not malignant tissue, but certainly contributes to the cancer ecosystem. And what we're learning is that many of these elements beyond the tumor cell expose vulnerabilities that we can take advantage of in beating back this cancer. So certainly the tumor cell remains first and foremost, but the genetics around both the tumor cell, but also the inherited genetics of the host, and importantly, the immune system and many other things issued here are all part of the cancer ecosystem. And what I'd say we've learned over the last 5 years is that these all represent new vulnerabilities that we can target. And the immune system probably represents the poster child of that. And here at the Abramson Cancer Center, we've been at the epicenter of driving that forward. And we call it the Cancer Immuno Revolution. And we'll talk a little bit more about that this afternoon in a cancer session. But the fundamental concept here, and I agree with Kristen, an image is worth 1,000 words, right? This is a T cell attacking cancer. So this is the basis of what's been going on, what you're reading in the New York Times and in Science Magazine and everywhere else, that T cells can attack cancer very similarly to how they attack viruses. Some of those principles are exactly the same. And we learned the molecular mechanisms behind that and we're able to target. And amazing things have happened with FDA approval after FDA approval. The way we take care of cancer patients will never be the same again. We played a large part here at Penn in discovering, driving forward the basic biology, and then doing the translational work to bring forward chimeric antigen T-cell receptor therapy to treat cancer. And amazingly, it worked, as shown here by FDA approvals of CART-19, for which we partnered with Novartis, and they called it Kymriah, so that's the name of the drug now. It was approved in August, as John mentioned, for acute lymphoblastic leukemia for both kids and young adults, and last week for non-Hodgkin's, certain type of non-Hodgkin's lymphoma. In each case, we thought we would celebrate because we felt, even down to the parking attendants, an ownership about what was accomplished in changing the way the first cancer gene therapy ever. And on the left is that flash mob John alluded to. It got so big I had to climb on a coffee stand to organize it. Dark blue represents the adult side here. Light blue represents individuals from Children's Hospital. We did this together. Madeline Abramson's in the middle of that. Amy Gutman's in the middle of that, and so on and so forth. Last week, we had a slightly different thing. We had a button flash mob where we told people, you know that T-shirt we gave you over here? Well, go get it out of your drawer, wear it again. It says an amazing thing happened, and get a button that says now for lymphoma too. If you like that idea and you want a button, it's right here. I feel like a Barker. Down the hallway is a big basket of them, and you can go back to Boston and put this on, and that would be awesome. You know, one thing I would say, looking at this, 2 points. One is, we talked about basic science. This is a 20-year overnight sensation. Don't think that this was an accident. Carl June is one of the most amazing T-cell biologists there is. I'll put him up against anybody in the country of understanding how T-cells work. He figured out the wiring, and the therapy was made. The second thing is the impact is amazing. We were talking about this yesterday. The rapidity to which this is becoming standard of care is amazing. There are literally hundreds of clinical trials all over the world, and the number of companies dedicated to this, and they all come to see us, is increasing several dozen now, really changing things in a way that's quite unprecedented. Let's talk for a minute about the future. So, you know, for every new therapy, there's immediately patients who don't respond, and we take that extremely seriously. So how can we make CAR T cells work better? Well, first and foremost, I think always fundamental is to understand what are the resistance mechanisms. We can celebrate the patients who are cured, you bet we do, but what about those patients who don't respond, and there's still too many of them? Well, we've begun to break that code. So we understand the target that we use to engineer these T cells, using a lentivirus to go after the CD19 molecule, which is exposed on the surface of hematological B-cell malignancies. That's not a necessarily important molecule for the lymphoma or for the leukemia. That tumor cell can just downregulate it, and that's what we're seeing, loss of that target. And we've been able to describe that in a number of high-profile journals as a resistance mechanism. Last week, to take this a step further, casting our attention away from what the tumor cell does, but to what's happening to the host, investigators published in Nature Medicine here from Penn and the Abramson Cancer Center that it's the patient's baseline immune competence that predicts whether or not they're going to respond. And what that means, for those of you interested in immunology, is that if a patient comes to us and has a robust memory cell compartment in the T cells, they will respond. If they don't, perhaps at the throes of chemotherapy, at the throes of the tumor cell, at the throes of other environmental factors or genetic factors, they don't respond. And we were able to predict in a cohort of about 20, 25 patients with 100% accuracy, would they respond, understanding the immune health of the patient as opposed to what's going on in the tumor cell. This is what I mean by the cancer ecology. And of course, there are mechanisms of tumor immune escape. I just heard one of our hematology oncology fellows yesterday at a research in progress, a CRISPR screen, and an amazing effort to find out how the tumor cells change themselves. How are we going to overcome resistance? You know, we need more targets besides CD19. They are rolling out. You will see all these developing. Targets in myeloma, other targets in lymphoma, and certainly we need to move into solid tumors. There's another revolution happening with checkpoint antibodies, these antibodies that block pathways such as PD-1 and CTLA-4. We've begun to combine those therapies. A recent report showing an amazing result in a refractory patient simply by adding anti-PD-1 to CAR T-cell therapy. And what you'll see, and is in the news, and of course you know Carl June was named by Time Magazine as one of the most 100 influential people in the world, and this year is so-called double engineering. So use engineering to confer a specificity to the T cells, but also use CRISPR technology to take away, for example, PD-1. And that clinical trial has begun here at Penn and has received a great deal of attention. And we plan on charting new territories. I don't have time to go into it, but we're looking to make these CAR T cells more than a patient-specific, right? We make each one from an individual patient, but to find a way to make them off the shelf and universal, to move into solid tumors like breast cancer, glioblastoma, I could go on, and to actually target mutations. So another hem-onc fellow has found a way to use CAR T cells to target the mutated KRAS protein, which would really be an amazing way to confer specificity. I have a few minutes to think to the future if it's okay. You know, this guy founded the place, and Ben Franklin, he did some other things in his life, but he did found the University of Pennsylvania. And if you go to the gym, you have to go down this sidewalk, at least the way I go, and it says an ounce of prevention is worth a pound of cure. And I think I've stepped on it 100 times until finally one day I looked at it and thought, you know, that is pretty good advice. So the last immune revolution was immune prevention. Polio vaccine, smallpox wiped out. So what can we do now that we understand how T cells work? What can we do? Well, we thought maybe there's a way to develop a vaccine to prevent cancer. And there's a group of individuals working here to develop what I would call a smallpox for cancer. Not to treat people who once have cancer, but can we vaccinate and mobilize the immune system before a patient is a patient so that they don't get cancer at all? And as we become to understand patients at risk, whether lifestyle or genetic reasons, we can offer potentially such therapies. Well, I won't go into the details except to say we again relied on our strengths, which is gene therapy, and partnered with a company that had licensed some Penn technology and designed a therapy which involves a DNA vaccine and a cytokine, all delivered as a plasmid DNA, with a very cutting-edge biophysical approach to use electroporation in vivo to deliver this vaccine to patients. And this trial, proof of concept trial, has begun. It just completed enrollment, and we're now beginning to study if this is possible. The setting of this trial actually was individuals who have cancer. We had operated on them. They were in remission, but unfortunately, we knew that they were at high risk. So these are patients with pancreas cancer, breast cancer, ovarian cancer, at high risk for relapse. So we let them get all the therapy they need, and then we said, would you like to join this clinical trial where we will vaccinate you against a target called telomerase, HTERT, to form an immune response against a very fundamental molecule in the cancer biology, and to try to prevent cancer from coming back. And it was a clinical trial. Here's some of the details for those of you who do this kind of thing. It was a multi-institutional partnering with all sorts of institutions, including others in Philadelphia. This is what it looks like. It's a little bit more than a flu vaccine, but has that sort of outpatient quality to it. And we're beginning to see, was it safe? We think it is. Was it immunogenic? It looks like it. Does it work incredibly well in other animal models? It does. And if everything adds up, we hope this year to bring this technology not to patients, but to individuals at risk. And our number one partner in that will be the Vassar Center here at Penn, who follow a great number of individuals who have mutations, germline mutations in BRCA1 and 2, and are at an 80% lifetime risk of cancer. We have some grants that have funding this, and our patients and individuals who are followed by the Vassar Center are already sending us emails saying, when you're ready, let me know, I'll sign up. So that's the kind of partnerships we have here. Now, speaking of the Vassar Center, and I'm almost finished, not at all do we think that immunology is all that's going on here at the Abramson Cancer Center. We talked a lot about PARP inhibitors today, and in fact, the FDA approval for PARP inhibitors for breast cancer for individuals who carry a BRCA1 and 2 mutation happened in January of this year. It was largely based on work from Susan Domchek, who worked with a national group, including many other cancer centers, published this New England Journal of Medicine article a few months ago that then the FDA looked at and felt the data was significant to lead to the FDA approval. These are pills, for those of you who don't know, that patients treat. It outperforms chemotherapy, and massively. And interestingly, for triple-negative breast cancer, which is the most dangerous type, this represents the first targeted therapy because 10% of triple-negative breast cancer patients have that on the basis of a BRCA1 and 2 mutation. So this was an amazing effort, as I said, led by Susan, who directs the Bassler Center. If you came in PCAM this morning, this is the sculpture you see going up the escalator. Take a look at, that's a molecularly accurate sculpture of BRCA2. Yeah, I love it. I see patients stop and take its photo, and then I take a photo of the patients taking a photo. That's the DNA. It's being repaired. This is BRCA2. Which was, our faculty member helped Barb Weber when she was here, helped clone and discover BRCA2. So it goes way back. The point I want to make, I'm sorry for taking such a long time to make it, is that is there a way to combine this genetic targeted therapy with immune therapy? Absolutely. The data in the lab indicates that these things should go together around damage done to tumors and how that exposes them to the immune system. In fact, studies led by a number of folks here, including a young investigator, Kim Rice-Binder in the Cancer Center, is bringing forward olaparib, which is the name of the drug, with other immune therapies, and we're very excited about that. So we join the Penn revolution. Later today, we'll talk about it. We're really focusing down, and I appreciate your attention. Happy to move to the questions.
So I'll invite our panelists up front. For those of you who may not know, Susan Domchek, who did those PARP1 clinical trials leading to the FDA approval for breast cancer, has a very supportive husband, who's Bob Vonderheide here. So I don't know if you have a conflict of interest in putting her successes. It's a powerful team. I'll begin with just a question or two, and then because our time is short, I'll open it up to the audience. If I can just begin, though, by asking Kristen, if you can just give me an example of the types of events. You were talking about electron microscopy, and of course, electron microscopy itself has been around for a long time. Clara Francini Armstrong here is a member of the National Academy and showed how muscles work by using electron microscopy a number of years ago. But there has been a revolution recently in cryo-EM, and if you could just give us an example of what's different. What are the kinds of things that you can see now that you couldn't see with traditional electron microscopy? I think that might help make the point.
So is that better? Yeah. The real difference is that we're now able to get much, much higher resolution. And so in the past, much electron microscopy was done looking at tissue, but not at the atomic level. And so advances in the physics of the microscopes, as well as the cameras, have allowed us to get much higher resolution and see atoms instead of just cells. And I think where we're really headed in this field is to not only be able to look now at atoms taken out of cells, but to see it in cells themselves. And so there's a new frontier, cryotomography, where people can drill down into a human cell and see at the atomic level how these machines, how BRCA, how PARP1, et cetera, how they look bound to the DNA in their activity within the human cell. And that's really tremendously exciting. We just had someone sign yesterday who's going to come here to Penn, who's able to look at pathogens, microbes that have infected inside of human cells, and he can watch those microbes interact with our host. I think this is tremendous for the future.
It's really a change in cell biology. I mean, we're really getting to the point where we can see molecules moving within cells. at the micro scale. About a decade ago, or maybe almost 2 decades ago now, there was a lot of national press about the valley of death in research, the great divide between early bench research and translating it into really making new drugs. And I hope you've gotten a sense that I think Penn's leading the way in getting across the valley of death. I think we built the infrastructure here, the culture here, and have the expertise here to not with just one-offs, but again and again, be really translating the most fundamental discoveries to affect patients. And I have the feeling in my own lab that the kind of things I do with mice and in a very basic science laboratory is not for the far away, one day maybe affecting patients, which is how I felt most of my career. I now have this urgency that we can turn things into real therapies, and we have the infrastructure to try it in patients, and we can see if it works. And just as an example, we're using CAR T therapy in my lab in animals to try to remove the fibroblasts, the scar tissue, from hypertensive hearts that cause diastolic dysfunction and relaxation problems. And, you know, quite dramatically, you can give a T cell targeted to the fibroblasts and wipe away the scar tissue within these hypertensive models. And, you know, it's quite clear that if that holds up, we have the infrastructure to do the trials necessary to show that that's a therapy. We have an eager questioner. Maybe, do we have somebody who can bring a microphone? There, yeah.
All this has to do with translational medicine, of course. You haven't mentioned your world-renowned proton therapy, radiation oncology, which is really, you know, one of the best in the country and the world. How is this all translated? This is at the molecular level. Now, I think the next frontier is going to be at the nano level, which, you know, which is much more infinite. But what are you doing in proton therapy and radiation oncology as you translate all this stuff to that department?
Yeah, it's a great question. Bob, perhaps you'd like to comment about insights from proton therapy.
I'm really pleased that you brought it up. I mean, it's a crown jewel, radiation oncology here at the Abramson Cancer Center and Penn Medicine for all the reasons you said. Proton therapy is a major effort here. We were among the first to do that. And there has been a great deal of effort to understand the capabilities and in fact, the limitations of proton therapy and to bring it forward to standard of care. Our observations are that there are an incredible number of situations where proton therapy outperforms photon therapy. You'll be seeing that data roll out over the next year or so. You know, in terms of your question of how we're going to advance that envelope any further, what has been amazing, actually, is up on the 8th floor of this building are a group of immunologists and radiation oncologists. They have their labs next to each other. They started talking, and they said, you don't suppose radiation could be like an immune sensitizer? This has turned out to roll out to a major concept called RADVACs. And the synergy that we're observing between radiation therapy and immune therapy is very, very powerful. And to take that step one further, the use of protons as the immune sensitizer seems to be particularly strong. I think we'll be the first in the world to show that. At the same time, we're bringing proton therapy to the community. We're using it as a cutting-edge tool to advance this type of transdisciplinary support. Really pleased to be able to share that.
Can you just very briefly give the type of example of RADVACs of what you mean by that?
So the idea and the current clinical trials, we have 5 or 6, is that a patient who would receive a checkpoint blockade, such as anti-PD-1 or CTLA-4, in the early stages of giving that treatment, we choose 1 or 2 tumors, isolated tumors, and deliver hypofractionated photon therapy to that one tumor. And what we observed, and we showed the mechanism in mice, that releases antigen and an inflammatory response at that local site, which acts as a vaccine, in fact, exactly as hypothesized, brings in T cells that then now understand what the tumor looks like, circulate, and find other tumors. That's the classic abscopal effect. Used to be observed by any radiation oncologist maybe once or twice in a career. You would radiate one site and see something regress. I see some heads. Now we can reproduce that 30% of the time by radiating one site and treating. So
It's a very amazing observation. And again, it's the close association between the clinicians and the scientists that figured it out. But, you know, these observations that patients were getting radiated here and their Mets were going away. And then looking back, and it was just those who were getting immunotherapy at the same time where that was happening. You know, somebody was smart enough to have a conversation about it and put two and two together and have a hypothesis about why that might be happening. So it was a great case of why academic medical centers work when you get people together. Another question.
I wanted to ask the three of you, none of you mentioned it, but I took care of newborns, and a particular field which has become more and more important, which I just saw a frontline story on, was the human biome, which may be involved in all kinds of diseases, in keeping ourselves healthy, in starting off human newborns. If they have a proper newborn, they can remain healthy for the rest of their lives. I wonder if anybody's investigating this and using it in their fields at all.
Did you say the microbiome?
Yes.
Yeah. Anybody want to comment? It's a big area of.
it's, I mean, you're right to point it out. The influence on health and disease of the microbiome is huge and becoming understood more and more. We have a very large effort. Some of the best people in the world are here at Penn. This has all been made possible. Our eyes have been opened because of next-gen sequencing that we can detect. Amazing things of how the status of the microbiome is influencing response to therapy. I keep talking about immunology, but you know, that is headline news, right? That you can now potentially predict who's going to respond to certain therapies. But it gives us something that's manipulatable, right? So we're already doing, believe it or not, you know, fecal transplants to treat people who have C. diff, for example. So in terms of, it's gone far beyond diagnostic and prognostic value. Now it's moving into therapy. And I think it goes way beyond cancer, actually.
Enormous
Diabetes.
area of focus here and at many places. We have a center that spans between Penn and Children's Hospital that focuses just on the microbiome. And we just, Kristen's reminding me, we just hired an assistant professor who's a chemical biologist who has peptide libraries that he synthesizes that can basically knock out any specific component of the microbiome to look for effects. And I'll just mention one result that captured my attention and was actually written up in the New York Times from Mark Kahn here, who showed he had mice who have a genetic predisposition to cerebral cavernous malformations, vascular malformations of the brain that cause strokes, and he's worked out the molecular pathway of the genes that cause this in kids and in animals. And when he moved to this new building, he brought his mice with him to the animal facility here, and all the mice stopped getting cerebral cavernous malformations, even though they had the genetic predisposition. And he, you know, Mark is a serious scientist who would not let this go. You know, he would track it down to the last nth degree, move mice back and forth, and it eventually did fecal transplants from the mice that were in the other building to the mice in this building, and showed that it was the microbiome in their gut that was necessary for them to develop bleeds in their brain combining with the genetic predisposition. And it's actually the first example of a anatomic abnormality, if you will, being affected by the microbiome. So the degrees to which it's going to affect all aspects of medicine, you know, are just almost beyond your imagination. Yeah.
Ralph Christeller, PGY 64. I'm compelled to make a comment. The practice of medicine is really very easy, especially if you're not a physician.
Thank you. We have other questions?
Hi, you mentioned the vaccine for cancer. How much is that running to administer to one person relatively, or is that still, I mean, is it super expensive? Like, what's the amount of it being ever brought to market?
So it's experimental. formulation at the moment. And the DNA vaccines have not been, they're not FDA approved, so we don't know how it'll be marketed out. So by the way, for our patients, it's free, right? There's hope that as this comes forward and will be FDA approved, if the data justifies, plasma DNA is really cheap. This is not protein chemistry, this is cheap stuff. And so there's hope that it will be quite affordable, actually.
But your question, if I may, your question brings up an important point that's in the press a lot these days, and that I think we're all concerned about, which is the cost of new medicines. And the immunotherapy developed here is a prime example of a extremely effective new therapy that costs an awful lot. And can we afford it as a society? Can we distribute it to all parts of the world and needy people, at this exorbitant cost? May be worth a comment or two. I'll just convey, first of all, it's not our fault. Novartis sets the price, and we have no say in the matter at all. Although we could come back to that. There are some academic institutions that are now, as part of their licensing agreements, are putting in clauses about what pricing would be down the road. I'll also say that new therapies that are really new are always expensive until they become more common and we work out the kinks and we figure out how to do it more cheaply. And that's moving incredibly quickly. If you have a chance to walk through the GMP facilities, the cell production facilities that were built in this adjacent building upstairs, that's where patients' blood are brought and cultured and transfected and grown and expanded and then brought back to the patient within the hospital. Is an enormous infrastructure that had to be paid for and built, but it looks like the Model T, if you ask me, of manufacturing facilities, where there's a big room for every patient's cells that are individually being grown and prepared. And we know that that's going to be miniaturized and mechanized over time. But we have to start somewhere, and there aren't that many patients who are approved for the present therapy. It's not as if it's millions and millions of people that they can sell the drug to. So some of my comments. Bob, do you want to make any comments further?
Well, it's, the rubbers hit the road because not just Kymriah with now 2 indications, but Yescarta has an indication. We are delivering these therapies as a commercial product in the Hospital of the University of Pennsylvania this week. I mean, we've been doing it. And, there's all sorts of considerations around a drug that costs $475,000. So now, let's think about that. For one thing, there are all sorts of alternative pricing models. Novartis is on record saying that in acute leukemia, if at 30 days it hasn't worked, it's free, right? So that's never been a type of assurance. We'll see how that plays out. The other thing is, if it works.
The good news is it's free. The bad news is it didn't work to cure your leukemia. But
yeah, it's total bittersweet, of course. So the other aspect is that there is, you got to take into consideration the total cost of taking care of that patient. And if it's possible that a single infusion of that therapy then offsets that patient having to get a bone marrow transplant or 3 more years of chemotherapy that causes hospitalizations. Actually, when folks have begun to add up the cost, the total cost, of this therapy versus what it would take if it didn't exist, it starts to get more reasonable. And at the same time, we have no way to get these other costs down. We have no way to get other costs down. We've been trying for 20 years as a country, and that's not working so well. But there are ways to get CAR T-cell therapy down. John mentioned a few huge efforts, and this is where we're going to have an advantage because we have a School of Engineering and a whole university, nanotechnology, and by the way, we have a 20-year head start on everybody else thinking about it. And I think the cost will get cheaper, faster, robotic-driven, these sorts of things that will hopefully go down.
I think many of us predict there'll be a universal CAR, an off-the-shelf product that doesn't have to be your own cells taken out and put back in. That would, in one step, make it enormously less expensive. There was a question up here somewhere.
So my name is Jonathan Berry, PGY 83. I'm a cardiologist, don't know a lot about microbiomes, although I hear about it a lot. So just expanding on that, what's the importance of the epigenetics and nutrition and our environment and how that interacts with immunology and biology and controlling neoplasm cancer?
Well, do you have an hour or two to? Anyone want to take that one on? They're looking at me, I'm afraid.
Were you asking about radiology? Because we could,
Yeah,
we don't mean to be flippant. I mean, every area you mentioned is cutting edge stuff, right? So epigenetics, you know, you know, this is now moving us beyond the genome. And, you know, we have amazing frontier investigators here. Shelley Berger last week was inducted into the National Academy of Sciences because of her pioneering work in understanding epigenetics. In fact, she has for many years been focused on the tumor cell. But again, in a hallway conversation or they had lunch together, she and John Wary, who directs the Institute for Immunology, got together and said, don't you think epigenetics should control immunology? So she shifted her attention, taught John the techniques in his lab, of course. And about a year ago, they published 2 science papers back to back saying that a resistance pathway for immune therapy is driven by the epigenetics imprinting, unchangeable imprint of the T cell. Amazing. So that's just an example to say our eyes are wide open and we have such an advantage here because we talk to each other and we work together.
and that pattern of epigenetic marks could help to predict who would respond to checkpoint inhibitors. So clearly a major part of what we're thinking about. Maybe I can just extend the question. Your question is broad and makes me think about the sort of dramatic changes in biology that have taken place recently. And again, it's sort of fun, especially in my position where we're trying to think about how to lay the bets for the future. What are the 20-year bets for the next revolutions that we can think about? Some of the fundamental things I was taught in medical school have to be rethought. The idea of terminal differentiation of a cell. If we can reprogram any cell with 4 factors to become an ES-like or IPS-like stem cell, it means that no cell is necessarily terminally differentiated forever. Couldn't it go backwards once in a while in nature? And when we look, it turns out there's more wobble, like your molecules shaking and moving in place. There's more movement between lineage positions of cells than we thought happens normally in nature. The primacy of DNA, making RNA, making protein, is being rethought. RNA itself has all these functions we didn't know about before. The DNA sequence in all of our cells isn't quite as stable as we always thought it was. And there's all these mechanisms to constantly break it and repair it. And sometimes to break it and repair it with intended mutations in the sequence, in our somatic cells, and maybe even in our germline cells. So, some recent work on how the immunoglobulin switches take place to make variable regions in immunoglobulins, the VDJ recombination events, have taught us now that there are sort of purposefully imposed mutagenesis that takes place in the variable regions that probably helps B cells have more diverse and appropriate antigens that they can recognize. But it doesn't happen everywhere across the immunoglobulin gene. It happens at specific regions. So random mutagenesis isn't so random. These sort of basic terms of biology make us have to think a little bit differently going forward. And in a way, it gets at your question about how all these different areas that are coming forward, affect one another as we think about how they might affect diseases. Any final questions? I think we're close to our time point. We've stayed on time, and I want to thank our panelists for that, and thank you all for your attention today.